Thank you for joining with us in today's special event. This lecture is entitled Discovering the Metabolic Roles of Hydroxysteroid 17-beta dehydrogenase by Genetically Modified Mouse in Vivo is presented by Doctoral Programs in Biomedical Sciences, Faculty of Medicine, Universitas Indonesia, and supported by Visiting Professorship Program DPASDP Universitas Indonesia. And already here with us is Prof. Asmarina, the head of Doctoral Program of Biomedical Science. Selamat siang, Prof.
Siang,
And
ya.
we would like to extend a special welcome to our guest of honor, Professor Matti Poutanen from Institute of Biomedicine and Turku Center for Disease Modeling, University of Turku. Good morning, Prof.
Good morning.
And I also want to say welcome to all of the students and participants to join us today. Before we start the lectures, maybe I will ask Prof. Asmarina first to give us a short speech as the head of doctoral program in biomedical science. Silakan, Prof.
Okay, thank you all here, especially our guests, our honor guest, Professor Matti Poutanen, that want to give us a special lecture today, this afternoon, and at the morning in the Turku. And also for the student that has already attended here in this meeting, in this guest lecture today, even. So I have also give thanks to the Pak Dwi Ari Pujianto, our lecturer in doctoral program in biomedical sciences, that introduced Professor Matti Poutanen to our study program. So I would like to give an honor for Professor Matti Poutanen for the willingness to give us the special lecture, and also for the willingness also to supervise our student Lulu. So Professor Matti Poutanen will be as a co-promoter from our student Lulu Yunaini that will go to the proposal examination next week. In this evening, we can hear so many information in transgenic animal results from Professor Matti Poutanen, because Prof. Matti Poutanen has a long research collaboration with Pak Ari, Dwi Ari Pujianto, and Pak Dwi Ari Pujianto has get PhD degree in Turku University under supervision from the Prof. Matti Poutanen. So we can hear the guest lecture, the nice topic today, especially for our medical student, biomedical student, because here is the important topic as biomedical sciences student here. So I think it is my speech for this introduction. Prof. Maybe Dr. Haimi Saida will also give a short CV from Professor Matti Poutanen. Thank you for all you here for this guest lecture today. Thank you very much, especially for Professor Matti Poutanen.
Thank you, Prof. Asmarina. And we are going to the main event, but before that, let me introduce Professor Matti Poutanen by reading his curriculum vitae. Let me give a share screen. Just a moment. Okay. So Professor Matti Poutanen is a professor in Department of Physiology, University of Turku, and director of Turku Center for Disease Modeling in University of Turku, Finland. And he is a senior scientist lecturer, Institute of Medicine, the Sahlgrenska Academy in Gothenburg, Sweden. He got his Master of Science in 1987 from Department of Biochemistry in University of Oulu, Finland. And he got his PhD in 1993 from Department of Clinical Chemistry, also from University of Oulu, Finland. And he became an adjunct professor in 1997 in biochemical endocrinology from University of Oulu, Finland. His research is mainly focused on sex steroid synthesis and actions, and his research group has been internationally in a key positions in providing evidence for the concept where the ligand concentrations available for steroid hormone receptor binding is markedly regulated by the target tissue metabolism. Sorry, can you please turn off your microphone? Thank you. Term as endocrine steroid hormone actions. And as of April 2018, he already has 6,270 total number of citations, with average citations per article is 26.8. And he has H index of 46 with 182 original peer-review publication. He is also a co-founder of Forendo Pharma that focus on hormonal therapies for endometriosis and male hypogonadism. Okay, and that is Professor Mati Pautanen curriculum vitae. I will ask for all of the audience to turn off the microphone because we are going to the main event. If you have any questions, you can write it in chat box and we will discuss about it after the lecture. So to Professor Mati Pautanen, the screen is yours.
Okay, thanks for the introduction. Is everybody hearing my voice?
Yeah.
And the screen is there?
Yeah, thank you.
Yeah, good.
Okay, thanks for the kind invitation and introduction. So it's my pleasure to be with you and really support your activities for PhD training. We just had such a nice time with Harriet Pujante years back, and then it's nice to provide also help for your training program. So these are two pictures from the two cities I'm really currently working with. So this on your left side is the river in Turku, where my main lab is, and as was introduced, I also have activities in University of Gothenburg in Sweden. So it's also on the front side, for the waterfront, and it's very nice environment as well. But unluckily now during COVID pandemic, I have not visited the place in last year at all. So that's really sad. But the topic today really is about the HST17 betas. It's the enzyme family I will talk about. And as I knew that you in Aripujantardos are planning to start working with genetically modified mouse models, and we have done that for years. So I thought I will give some, aspects and views how we have been using those animal models and why we have used a lot of in vivo models to understand the biology of these enzymes. And now, of course, entering also to in clinical trials with inhibitors with some of these enzymes, and hoping at some time later, maybe in the future, also have some benefits for the patients. Currently, none of the inhibitors are with these enzymes are not used in clinical practice. So really, to start with that, so I will refer to this enzyme family as HST17 betas. So this is a shorter version of the word of this enzyme family. So these enzymes actually are, they belong to a larger family of enzymes called short chain dehydrogenase reductases. And what these enzymes do actually, so they do the hydrogen transfer, and why the enzymes that we are now working with, the nomenclature actually came from the fact that they are converting the 17 ketoforms of these steroids. This is the ketosteroid on position 17 in steroid, and it converts that to 17 hydroxyform. Just putting one hydrogen atom there in this molecule actually brings this molecule far more potent. So these steroids, classical steroids, as you really see, estradiol and testosterone, they are 17 hydroxyforms, and these are like 24 more potent hormones than the 17 ketoforms. So the idea when we start to work with these enzymes was that, okay, we have here a switch, enzymatic switch between inactive hormone and active hormone. And this was the kind of basis for our studies and why we entered into looking on the enzyme family and these activities. And therefore, it was an interesting concept because these enzymes are also expressed in peripheral tissues. So the idea was that, okay, maybe they are doing like a switch, active and inactive ligand for steroid receptors as that are in the nucleus, as many of you know. So many of these HSD enzymes are really, they are dimers or homodimers or tetramers in the active stage. So this is actually where the name comes from, these enzymes. So it's because of the 17 beta, hydroxy form conversion and ketohydroxy conversion. So, but anyway, at the same time, when all this, the whole genome has been sequenced, so it came very much clear that among these SDR enzymes, where these HSD17 betas are, so in this larger family of enzymes, which is about 70 enzymes in humans, so they have a lot of other activities around, so that some of these enzymes that are in this family are using substrate such as, in addition to steroid hormones, has substrate as prostaglandins, retinoids, lipids, and even xenobiotics. So what is common to these enzymes are that the catalytic mechanism is similar, but their substrates are different. So, and that's actually forming this group of SDR enzymes, where this small group of 17 beta HCs, these are, and which we have like 15 to 14 in humans. So the interesting thing here then came when you look on these HSD17 betas and you scatter them around the bigger family of SDRs. So they are not forming their own subcluster here in this bigger family, but they are scattered around this big family. And that already gave us an idea that, okay, there's two possibilities, that maybe all of these enzymes, even they are named based on the activity of steroids, maybe they are not steroid dehydrogenases per se, but they are doing something else in vivo. Or then this other possibility was that maybe the primary sequence actually is not folding, is not determined for the three-dimensional structure of the protein, that if all these enzymes are primary steroid dehydrogenases, so why are they so different in their primary structures? So this is really something that we have been trying to understand, the physiology of enzymes in vivo, because you all know that in vitro we nowadays have very sensitive systems, and you can basically, for enzymatic reactions, you can put a small number of substrate and you can incubate 2 hours, maybe 2 days. A lot of people do that 2-day incubations, and then you see a conversion.
Oh ya udah kayak pas saya pakai 2 aja. Oh iya.
Please turn off your microphone. Thank you.
So yes, so at the same time, like in case of steroids, the half-life of the steroids in the body is much shorter than 2 days. So the sort of physiological importance of any in vitro reaction, if you incubate 2 hours, doesn't really make sense. So, but anyway, in the beginning, so we were interested on steroid metabolic synthesis and steroid inactivation, and the indications that we were looking for were breast cancer, prostate cancer, endometriosis, and trying to really see that how are these HSD17 beta enzymes involved in regulating these hormone-dependent diseases and also hormone-dependent growth and function, and what are really the physiological functions for these enzymes. And to challenge that, so we did a lot of, or have been over the years done a lot of knockout animals, meaning deleting the enzymes in mice and then looking what there is going on with the mice and trying to understand whether these features that we see are associated with steroid hormone action or are they associated on something else. So we were thinking that in addition to doing a lot of in vitro work, so we challenged the physiology of these enzymes. And I will just sort of summarize, what kind of consequences by deleting these enzymes in mice will, what will happen, and then some of the conclusions we can draw from those. And actually, it was very surprising to really some of the phenotypes that we got changing in the mouse physiology were quite surprised if you think that we were looking for steroid hormone action. And already dehydrogenases, they are doing something else, and this is something we discovered by using in vivo models. The problem with the in vivo models, of course, is sometimes, and it's often is that it's difficult to to discriminate the primary effect and secondary effect, and the strongest physiological effect will mask everything that is under that effect. So even if you are making a knockout animal, the animal is dying. So anything that might have to do in adult animals, we are now doing a lot of point mutations and tamoxifen inducible knockouts and so forth. But some things that I just want to go through with you. And first thing that what we did is that we worked on HST17 beta 1. And this is an enzyme that was cloned, first HST enzyme that was cloned actually in our laboratory, maybe 20 years back. It's highly expressed in ovary. So this is the immunohistochemistry in human ovary. The enzyme is expressed in granulosa cells throughout the follicular genesis, and it's also expressed to some extent in the corpora lutea at the early phases and not in the later phase. So just to let you know that this enzyme activity is essential part of estradiol synthesis. So basically, if you start the classical steroid synthesis from cholesterol, so there's no way to get estradiol without having this reaction to take place. The question is then, what is the enzyme responsible for this activity? So we were hypothesizing that it's type 1 enzyme because it's very highly expressed in the granulosa cells in those cell types that are involved in estradiol synthesis. So, and in a mouse, it's about the same. Steroid synthetic pathway is the same in mice and human. So what happens when we delete this enzyme in mice is shown here actually. So what happens is that if you look on the enzymatic activity that we are looking, the activation of eastern estradiol, the white bars are the knockouts and the black bars are the wild types. You can appreciate that most of the activity is gone in the ovary. So telling that this enzyme really is the key enzyme producing estradiol in normal setting. But what was surprising for us to identify anyway, that even you kill this enzyme in mice totally, so basically you kill the activity into 90%, still the ovary are producing normal amount of steroids. So there's very strong compensatory effect in the ovary that is able to produce estradiol even in the absence of this enzyme, which was surprising to us because we thought that this enzyme activity is essential for estradiol production. So that was a kind of lesson number one, that even it's very highly expressed. It's one of the main enzymatic activity in ovary concerning the E1 to E2 reaction. Still, in the knockout mice, you have normal estradiol concentration. The compensatory effect is very strong. So what we found anyway, that there was also effect in the ovary.
So,
Hilang ya?
what happened? The connection is broken.
Iya, Prof.
Yeah. So, ya, jadi kita tunggu ya. Mati potanennya keluar. Tuh. Pak Ari bisa ini apa? Kasih tanya ada apa?
Saya WA ya.
Oh ya, kamu bisa WA ya langsung ya. Oke.
Oke, dia minta itu lagi.
Itunya linknya.
Sebentar.
Okay, we are back.
Okay.
What happened?
Yeah, I don't know. I was thrown out, so maybe it was too tough.
Okay.
Yeah, so we are back here anyway. So thanks for all. We're just putting the.
Yeah. Okay. Never mind.
I don't know what happened.
The distance for us is so very, very far away. So,
Yes.
yeah. Some problem with our connection.
Yeah, maybe something went wrong in Europe in the lines. But anyway, so yes, so we were able to show that even we delete this enzyme in mouse ovaries, so we still have estradiol production, which was surprising to us. So the other surprise actually for us was that this enzyme, which was supposed to be an enzyme involved in ovarian granulosa cell estradiol production, was also expressed in testes. So we found out that it is expressed in Sertoli cells, both in mice and in human. And then we were really surprising that what would be the role of this enzyme in testes. And it's still unknown, but surprisingly, what we found out that here in your right side, the spermatogenesis is disrupted in these animals when we delete this enzyme. So somehow, when you don't have this enzyme in the testes, it's somehow the Sertoli, so the support for spermatogenesis is not fully committed. So and we don't know the mechanism at the moment, but this was a surprising finding, and we are trying to understand this further in our current studies because it was surprising. Is there a role for local estradiol synthesis in the Sertoli cells and activation that is important for Sertoli cell, supporting spermatogenesis? Nobody knows about that. It was a surprising finding. But one of the reasons that why we are studying this type 1 enzyme is that we were thinking that when it's expressed in peripheral tissues, it's one of the mechanisms how you can also make ligand activation locally in the target tissues. And in human, it's highly expressed, like in breast cancer and other peripheral tissues that are estrogen dependent. So what we did, we were kind of mimicking a situation that we overexpressed this enzyme in animals, in all the tissues, but then we took from those animals that had high expression of human HSD type 1, we took the epithelium from those mice and then transplanted that to a sister mouse, which was a wild type. And then we put a wild type transplant to the other side of the. So in mouse, when they are inbred strains, you can do the tissue transplantations, and they don't it's okay. Then the immunology is accepting the transplantation. So now we could look at what happens in mammary gland when there is HSD17-beta1 overexpression, but otherwise the animal is totally normal. So that was a very interesting model that we did. And interestingly, what we found out that when you have high amount of enzymatic activity, we just look then in wild type mammary gland, and then in those that we transplanted using transgenics, we could see that there's much more estradiol produced if you make enzymatic activity measurements. And what happened is that in these animals where in these mammary glands that were specifically overexpressing this enzyme, so they were starting to have small lesions. And mostly what happened was that this inflammatory reaction, taking place because this myoepithelium, which is actually surrounding the epithelial layers in mammary gland, was broken down. So just overexpressing the enzyme in the mammary epithelium, break down the myoepithelial layer, and make this kind of very strong, immunological reaction. And this reaction could be inhibited by HSD type 1 inhibitors, and we could also inhibit that with estrogen receptor alpha blockers, ICI, suggesting that this effect was estrogen dependent. And this was really interesting. And another interesting association with this is that we have seen that this enzyme is somehow expressed in invasive border of the mammary cancers in humans. And this is something we are now trying to understand, that does this enzyme, when it's locally expressed in the mammary gland, does it increase the invasiveness of the epithelial cells? So if that would be the case, so there will be some clinical implications also maybe for inhibited, using treatments for patients that have aggressive tumors, mammary gland tumors, breast tumors. So this is an interesting kind of translational aspects of these results that in target cell tissues. So this is really a kind of a summary where we are with the HSD type 1 enzyme, and it has been very fascinating to be in the field that there is a hypothesis that we have, other enzymes that are inactivating the steroids, and then we have other enzymes that are activating the steroids in the same family. And you can do this really in vitro, and in vitro you can see that this enzyme, like the type 2 enzyme, preferably inactivates the steroids to the low potent steroids. And if you again look on the expression of this type 2 enzyme in humans, so it's really seems to be co-expression in the situations that you would really tend to think that this inactivation is important. One example being in the endometrium during the cycle. These are controls and these are actually endometric patients, but this is endometrium during proliferative phase and this is secretory phase. So this enzyme that inactivates estradiol is really highly upregulated in the endometrial epithelium on the secretory phase. And its expression actually is negatively correlated with estradiol concentration in the tissues that we have measured with mass spec. So this was just an example, but there are also other examples to suggest that, okay, this would be really an estradiol inactivating enzyme. Okay, then the question would be, okay, what happens if you knock out these mice? Do you see an inactivation of estrogen action in these mice? And a short question is, no, you don't. But you see something else. And the most striking effect that we saw in these animals is actually that the placenta is, development is disrupted. So if you knock out the animal, the enzyme in mice, the placenta is not normally developed. And as a consequence of that, a lot of the fetuses are not growing normally. So nothing related to estrogen action was found in the knockout animals. And similarly, if you overexpress this enzyme in mice, so what happens is that you have, delayed eye opening, and you had also some changes in retinoic acid dependent genes. So our conclusions came to that okay, it seems to be that this enzyme is more involved in retinoid signaling than steroid signaling. Again, we have not really had time to go deeper on this, but again, suggesting to you that the in vitro data and in vivo data doesn't really match nicely, and the in vivo data gives, really potential other roles for this enzyme. And I think this is the common theme with everything we have been doing. And many of these enzymes, when you do that in vivo studies, you are not able to confirm the in vitro studies. And I think in that sense, the in vivo studies are very, very powerful, because they have to some extent, your in vitro data had to to translate to in vivo to be confident with your hypothesis. One enzyme that we have been working with, which clearly, you can confirm the activity on both in vitro and in vivo, is this type 3 enzyme. So now we are talking about type 3 enzyme, very similar to that I taught for the type 1. It's highly expressed in ovary for the type 1. Type 3 enzyme is the key enzyme in testis to produce testosterone. Again, as I mentioned, for the type 1 enzyme, in ovary, there's no way in the testis to produce testosterone if you don't have this enzymatic activity. And the question again comes, what is the enzyme responsible for this? The candidate enzyme was type 3 because it's highly expressed in the testis. Other reason why we're thinking that this is the candidate enzyme, and actually was showing that this is the enzyme, is that there are human mutations for type 3 HSD. And the type 3 HSD deficiency actually results to the fact that if you are XY patient or boy, so they are born with female appearing external genitalia because they are not masculinized properly, because there's no normal testosterone production in the fetal age. What happens to these patients or individuals so is that at the puberty, testosterone starts to rise. Very similar to that I showed in the knockout for the type 1 in the ovaries. Again, even you have a classical enzyme that is responsible for testosterone production, when there's enough precursors accumulating, other enzymes are taking over, and these actually individuals are masculinized during the puberty. But clearly showing that if you have deficiency in this enzyme, the fetal development masculinization is not normal. So can we replicate that in mice? And that was a question. And then we did a mouse knockout animals, and this is true. In this case, we could really replicate what happens in human with the mice. And the data is shown here. This is now knockout data that they have the anogenital distance is shorter, so they are feminized. They have later puberty. Their body weight is a little bit increased, meaning that they have maybe esthetization. The anabolic effect is maybe reducing. Testis weight is smaller, and many of the androgen-dependent tissues are smaller. So clearly, there is a lack of androgen action in these animals. And the puberty is delayed, and the testosterone synthesis is not normal. Very similar to that in the patients. Interestingly, also in endocrine system, it's very similar in mice in this case that, again, you can appreciate here that we look at testosterone concentration in adult animals. So it's normal. It's even uppers a little bit higher than in wild type animals. So again, similar to that in the patients, in an adult animal, you really get a normal testosterone because other enzymes are going to be involved in testosterone production. So endocrine disruption in these animals are very similar to that we see in the patient. And very similar in the patient, we have very sky-high androstenedione, which is a substrate for testosterone production. It's upregulated very strongly because there's a block from androstenedione to testosterone conversion in these mice, similar to that in the patients. So actually, the testosterone androstenedione ratio is a clinical marker for this deficiency in patients. And you can appreciate that this is the same thing in the knockouts, that it's very much different in the knockouts compared to the wild types. So the endocrinology is very similar in mice and human in this respect. Also, the fact that the LH is sky high in these animals, similar to that in the patients. So what we could now conclude that the endocrine environment in these knockout animals very well mimic the patients with the mutations, which are very rare in our societies, but they are actually quite common in Gaza region in some Arabic populations. And the interesting thing, what we could show in these knockout animals is that that the testosterone that is produced in an animal is, coming from the testis. And this was, in a way, interesting finding because a lot of the patient, reports are suggesting that would be peripheral conversion. So now this would be interesting to see whether that's the same in the patients that actually then finally they are, during puberty getting more and more testosterone. So is it really testis, dependent? We don't know that at the moment. But this is something that really came along. So to conclude here, that we could conclude that these mice, knockout mice, are very similar to that of the patients with this enzyme deficiency. They are undermasculinized, they have high androstenedione, the testosterone rises at puberty in both cases, they have lower testosterone to androstenedione ratio, and they have very, very high LH. Now the question is that could we use this mouse model somehow to look on what would happen or what are the comorbidities in patients if they are not treated, or during their symptoms. And one of the things that we are now following up is the metabolic syndrome and weight of these animals because they seem to be a little bit overweight. And the question is that do these patients then that if they are not treated, with hormone replacement and or surgery, do they have similar problems in the metabolic features? So this is something we are studying. But interestingly, anyway, we could really replicate the endocrinology in mice that we see in the patient. So that was a positive sign for us that at least in some of these enzymes, they really seems to have we can mimic our hypothesis, we can confirm our hypothesis in using knockouts. Story is totally different about HST type 7 that we have also worked with. And this is an interesting story because years back, it was expression clones from mouse ovary to be an enzyme that converts E1 to E2. It's expressed in corpora lutea. So basically you would expect that, okay, great, this is the enzyme that is compensating the type 1 in the ovary. As I showed in the beginning, even if type 1 is missing, the ovaries are producing normally the estradiol. So would it be type 7 then having this option? So this is still, I think, an option. But the problem with looking on this option is that when we delete this enzyme and looking forward for its role in estradiol production, we found out that actually it is very essential for cholesterol synthesis. So when you delete this enzyme, the mice are dying in utero because they don't normally produce cholesterol. And you know that cholesterol is needed for example, for the brain to develop. De novo cholesterol synthesis is important. So the brain damage will kill the animal. At the same time, also the cardiomyocytes didn't develop normally. So we could also show really the blockage in the cholesterol synthesis because in the knockout animals, some of the intermediates were the blue bars were really highly accumulating, and some downstream intermediates were lacking. So we clearly could show that these animals are lacking cholesterol synthesis, and that affects then on development of the brain and heart. And we were able then to show whether it has a role in ovarian steroidogenesis. This is something now we are working with, so because we have also Krylov model for this, so we can activate now for example, the enzyme in the ovaries. But still we have a problem because we know that cholesterol synthesis is essential for steroid synthesis. So if this enzyme has a double role, it's really very, very difficult to show in vivo because the lack of cholesterol synthesis most likely will mask whatever we could see in steroid synthesis. Very similar study actually case is for the type 12 enzyme when we have been deleting that enzyme. Again, it has been suggested to be involved in steroid synthesis, but when you delete the enzyme, we didn't see, what happened in these animals was that they don't grow at all. These enzymes is essential even for embryonic stem cell proliferation. So there's almost no organogenesis in mice if you delete this enzyme in animals. So it's very essential for organogenesis. It's also very sensitive. There's a dose, sensitivity in this gene because even in heterozygous mice, lacking one allele, we start to see effects. And the first effects that we see are in prostaglandin synthesis, without going into any details on this. So now again, we are in a situation that we are not affecting steroid hormone synthesis as shown here, but we are affecting, in this case, in prostaglandins. Again, we are in a situation that, okay, the nomenclature might not be right. This enzyme is not really a steroid dehydrogenase. This is working somewhere else. And the current understanding is that it's involved in fatty acid elongation. It was very interesting for us, to follow this a little bit, and that's why we did a Krelok system, and to be able to knock out this enzyme in adult animals, because if you knock it out in germline, as you say, I showed, there's almost no organogenesis. So it's very essential for the early life to appear in mice. So we got very fascinating results when we did an gene inactivation in adult animals. So we can do that by crossing the floxed type 12 HSD animals to ROSA 26. 3 mice that then introducing the knockout by giving tamoxifen into the animals. And this was very surprising results that we recently published, that when we give 3 days tamoxifen for these mice and then knocking out this enzyme, so the animals are reducing their weight very rapidly. In 1 week, they are losing 20% of their weight. They are losing almost all their fat. So fat tissue is gone, and animals are really getting, very, very thin. At the same time, some of the fat is accumulating in the liver. Some of the liver damage markers are upregulated. So very strong metabolic actions in 1 week when you inactivate this enzyme in adult age. And then we were following this a little bit more, and we could show that the food consumption went down, the water consumption went down, and then the question is whether this is that they don't eat and drink. Is it just that there's a toxic effect? That's one option. The other option is actually that now we have found out a regulatory system that regulates the feeding behavior in these animals. And that option is very interesting, because if you look on then the signaling in the hypothalamus, the orexigenic signals are really up and anorexic signals are down, meaning that the hypothalamus is telling to the mouse that you should eat, you are starving, but still the animal don't eat. When we started losing the weight in these animals, they are still happily walking around. And so we think at the moment it's not toxic effect that they stop eating because they are so sick. We think that they stop eating because there is some, signaling related to the dietary habits is disrupted. And this is very interesting because if you look on genome-wide association studies in human population, this gene has been linked to weight loss. And this is something we are now following. Could this be really one way to regulate the appetite and weight loss? And that's a very interesting and provocative idea that we are following at the moment. Finally, the last piece of information I will share with you is this type 30 that we recently deleted in mice. And why we are still studying this is an interesting thing that we, a couple of years ago, we published a paper and we came to the conclusion that this is really, we couldn't really figure out what's going on, but these animals, they get liver steatosis when we were deleting this enzyme in mice. It's highly liver-specific enzyme. Again, nothing in the steroid hormone action, nothing in the reproductive functions, but they something in the liver metabolism getting wrong. And then we just, wrap up the paper and we thought, okay, we will, forget this story because it's so complicated. But just, a couple of weeks after we had our knockout paper out, there came a very nice story in the New England Journal of Medicine that inactivation variant of this enzyme actually protects humans from non-alcoholic fatty liver disease. And then, it's a very strong research came along on in this topic, because there's a huge need to find new ways to treat, liver steatosis and liver cirrhosis. And just it was so fun because just as we were thinking that we will, forget this story because it's so complicated, the whole field was dramatically came in on the top of the hepatocyte biology because there's not so many so strong variants and Jiva studies indicating that the targets involved in protecting the humans for getting this liver disease. So that's why we are now following this very, very carefully now in many aspects, and even it still is complicated. So just to summarize this a little bit, I think fractionated story and all these different HSTs, just to summarize, as also was mentioned, I'm involved also in a small drug company where we are trying to understand and thinking how could use this information for the benefit of patients. So to summarize here that we have HST type 1 inhibitors, they are now in phase 1 and phase 2 trials. So they are very nicely tolerable for human. The question is that how efficient they will be is still a question, but we are working on type 1 inhibitors as indications for endometriosis, adenomyosis, and endometrial cancer. So the company, will mostly working on women's health at the moment. Type 5 HST, I didn't talk about at all because it's an ACARE enzyme. It's not on one of the SDR enzymes, but we have a preclinical project on PCOS and CRPC, prostate cancer, on this. As I just showed that this type 12 enzyme is really interesting concerning regulation of the weight gain. So we have to follow that path to some extent. And then we really want to follow also this type 30 NFLD and NASH field, but all this is in a preclinical phase at the moment. So only clinical study phase inhibitor we have is for the type 1 HST17 beta. Just to finalize, and thank you for listening and thanking all the people who have been working over the years before, earlier and later, and all the collaborators. And unluckily, I didn't share any data that where Ari Pujanto was involved. He was working more on reproductive field, where we have been working as well, in addition to the HST17 beta biology. So, but I personally have not been very active on that field anymore, even though in the lab there are other people very strongly working in a similar topic. So thank you all for listening.
Thank you, Prof. Matti Poutanen. What an amazing lectures. Now we are going to have a discussions about the topic that Prof. Matti has already introduced to us. Is there any questions? Maybe from Marina or from Pak Ari?
Okay, can I have a question? Okay. Amy?
Yeah,
Izin bertanya. Okay. Okay, Matti, thanks for the nice presentation. Concerning the effect or the role of this enzyme, hydroxylase, what is the name of the enzyme? 17 SST beta type 3 in male, for example. You mentioned that this enzyme also have a role in the steroidogenesis in male, and then there is also phenotype in male when you knock out this gene. So where is the expression of this 17 beta HCG type 3 in the gonad, for example? As we know that actually testosterone is produced in the Leydig cell, and then where is the expression, the highest expression of this 17 beta HCG type 3?
Yeah, I very shortly. I was trying to share, but anyway, so as I showed shortly, so it's expressed in Sertoli cells, and that's a kind of interesting story. It's not in the Leydig cells where testosterone is produced normally.
Okay.
And in a mouse, the Sertoli cells has been shown to be involved in steroidogenesis in fetal life. And that might be a little bit different from a human. So, and it was kind of fascinating that there's data, not from mouse data, but showing that Sertoli cells in mice contribute to steroid synthesis in fetal age. In adult age, it's Leydig cells also in mice. But as I showed shortly, both in humans and in mice, these enzymes is in Sertoli cells, not in Leydig cells where you would expect to have testosterone production. And that was really a surprising finding for us, because I thought that this would be something that is specific for mice and it doesn't translate to human. But we then got testis sections from our collaborators in Munich, and it was nicely expressed in human Sertoli cells. So now actually we are doing currently double knockout, type 1 and type 3. And then really to actually pinpointing, no, I had to, yes, I was talking about type 1 all the time, sorry Ari. Because of course type 3 is in Leydig cells as it should be. Okay. So that's clearly like in human. Type 3 is in Leydig cells producing testosterone. Okay. But surprisingly, there's also type 1 in the Sertoli cells, which we thought to be only expressed in ovary because it's estradiol producing enzyme. So the type 3 is exactly like in humans. It's Leydig cells, it's there, and it's important for testosterone synthesis. And as I showed, patients who are lacking the enzyme don't get normal testosterone synthesis. But the funny thing is that those patients or individuals masculinized in the after puberty, meaning that when you have a blockage in the late stage of testosterone production, the precursors are accumulating, and then when there's high enough of precursors, some other enzymes are taking over. And now we have tried to understand whether that's type 1 enzyme that takes over in the testis. Because as I showed, at least in mice, even in a lack of type 3, still after puberty, testis is responsible for the testosterone production. But the human data, people working with the patients were thinking that it's peripheral conversion. But we showed in the mice that it's still coming from the testis. And now we know that also type 1 is in the testis. So now we are making double knockout to see, can we diminish, testosterone production from the mouse testis if we have type 1 and type 3 both lacking. But type 3 is normally in the Leydig cells. So that's really like translating nicely to the human data.
Okay, very interesting. So for example, so it seems that these enzymes can be used as a candidate for therapy, for example, for these estrogen related disease, for example, endometriosis, breast cancer, or ovarian cancer, for example. But in the signaling pathway, actually the final product of this pathway is actually estrogen. And then for example, in the follicle cells, for example, estrogen is coming from testosterone by aromatase. So what is your opinion? Which one is more effective? Designing the therapy using this enzyme or aromatase inhibitor?
Yeah, you are very much right. Certainly aromatase is more effective because that kills all the androgen conversion to estrogens. And what we are thinking and hoping for, that actually using these HSD inhibitors, like for case of endometriosis, that the therapy would be having less side effect than aromatase inhibitor, because you don't want to give a young women aromatase inhibitor killing their bones and make them postmenopausal. It's not very nice for women. So we are thinking that if you inhibit type 1 enzyme, you still have estrogen around. You keep on some estrogenic action, but you wipe out the peaks of estrogen action. So this is my current idea. you are absolutely right. I don't expect that we are going to have the same efficacy that as we have with aromatase inhibitors. Concerning breast cancer, we are hoping that to have an adjuvant therapy that you could, sometimes there are people who are even resistant to aromatase inhibitors because it could be estrol sulfate pathway coming. There are also other pathways for in women that might bring these kind of estrogens from the long-term storages in adipose tissue that are then converted to active estradiol. But you are right, aromatase. But the problem with aromatase inhibitors are they are so striking, they are so strong. People are using that at least in USA for endometriosis and even GnRH analogs, but they are very, very strong drugs. I mean, for fertile women.
Okay, thank you.
So yeah, I would like also ask, Dr. Imi.
Silakan, Prof.
Okay, yeah, thank you for nice presentation, nice lecture, Prof. Mati Potanen. Yeah, I'm interested with your clinical study from the administration of the inhibitor of HHD17 beta 1, the isoform 1, type 1. So in your presentation, this enzyme play a role in the oocyte, oocyte development or in ovary, but you use this inhibitor to treat the endometriosis and also endometrial cancer. Yeah, how can we understand this role of the enzyme in the ovarian, but you use the inhibitor to treat the endometriosis and also in the endometrial cancer? So
yeah, I did have a very good question and really important to figure out. Yes, so our idea is that I didn't have time to show, but that's a topic of another lecture. But in the circulation, you have the estradiol ratio. I mean, you have and we have shown that this enzyme is expressed also in peripheral tissues. So we are thinking that without affecting ovarian synthesis, we are able to manipulate locally in the target cells the estrogen concentration by inhibiting this enzyme, because we are expecting that part of the, and we are studying that a lot now because we use mass spec methods to look on steroid concentration in the tissue and in the serum. And if you look on estradiol concentration, for example, in the endometrium during the cycle, it's much, much higher in the tissue than in the serum. And also estradiol to estrol ratio is changing. So we are thinking that part of the mechanism how the ligand regulate the steroid hormone action in target tissues comes from the local activation and inactivation. That the ovaries are producing the mainstream, but what the target cells are seeing is not only the steroid concentration that is in the serum, but they're also locally produced. And we are hoping that in the target cells we can manipulate the steroid levels by these enzymes. And I think similar to that in aromatase, it's so strongly expressed in ovaries, so we don't think that we can inhibit ovarian synthesis very strongly. But again, if you think about breast cancer, you certainly know that they are mostly postmenopausal women. And again, the situation is totally different. And then like aromatase inhibitors are very efficient for the postmenopausal women because it's peripheral activation. It's in the tumors and also in adipose tissues. Ovaries are shut down. Still, you have a low amount of estrogens around, and you're blocking that by aromatase inhibitors. So there, I think type 1 inhibitors really could play a role together with aromatase. But again, as I said to Ari, I mean, these likely are not, I would think that these compounds would be more like modulatory, not really like, on and off like aromatase inhibitors or GnRH analogs that are really shutting down the whole pathway. The problem, what we are facing and challenging with is that we, as I showed, these potential compensatory effects, which is surprising. Like in aromatase, there's no compensation. And I'm very surprised. You have one enzyme, you totally inhibit the estradiol formation from androgens. It's very surprising because it's so key reaction if you think about the reproductive biology. Why there is no backdoor pathway to produce estradiol from androgens? This is surprising. But yes, you are right. So we think that it's the target tissue metabolism that we are affecting. But let's see. But I think, as I said, in postmenopausal women, it's easier. In premenopausal women, of course, it's a little bit more challenging to really how much new biology we will find. So you're right.
Okay, one questions more. So yeah, with the animal model, yeah, we can evaluate the function of the gene, yeah. And so in this, concerning with this enzyme, there are 70 type of the enzyme, and if we have already made the knockout or transgenic animal with this gene, and we can find some effect, that surprisingly, you have already also found the surprisingly result with this animal model, knockout mice, and so on. So I would like to know how can we arrange the animal model lab? Yeah, maybe have you any suggestion that what is needed for the animal lab model for this knockout mice, because we would like also build the animal model lab in our university, in our faculty. But what should we do, to develop this animal model laboratory? Because with this knockout model, animal model lab, we can evaluate the function of the gene, and then the application of this result we can also apply for the treatment of this disease. Yeah, I mean, so maybe you have a suggestion what should we do to build the animal model lab?
Yeah, this is true. As you might know, even we didn't discuss. I have been running this unit for 20 years in Turku and have done maybe 1,000 transgenics and 100 knockouts. So it really depends on your wish and how much you want to invest. Like in our case, also in Finland, we have some rare diseases that nobody else is interested in studying. So we need to do ourselves the knockout mice and point mutations. So if you have situations like that, so it might be good to have the technologies around. But there's a lot of, at least like the model that we will discuss with Ari Pujanto, is that there are big labs who is making these for you. So the question is, do you want to start building them up, or is it enough to order them from collaborators or from repositories and building up first the facility that you... I would start by building up a facility that would have a nice animal facility, all the protocols that familiar with genotyping, and then you could just start maintenance the colony, because the key thing and the most important thing to start with is reliable PCR, because as you know that every single animal needs to have a social security number, as you see. I mean, if you start mixing your genotypes, so you mixed everything. So that's my key information to all my students. Never mix a genotyping, because whatever you do after that will be catastrophe.
Okay.
And we have seen that in my lab, even that people don't do proper genotyping, and then your results are, they are worthless. I mean, they don't mean anything.
Okay.
So having very nice protocols in animal laboratory, very robust PCRs, and for example, in our case, for every single knockout, we have at least 3-4 different genotyping protocols, because one day you will wake up and the old protocol don't work.
Yeah.
And you don't know why. It doesn't just work, and you have to change the protocol. So I think that's really the key, whatever you do. Then the second step is, if you want to start producing your own knockouts, so then you need a lot of facilities and expertise to now, of course, less, because now people are using CRISPR. So it's actually quite easy to make deletions with CRISPR. But we have found, if you want to make point mutations or certain specific deletions with CRISPR, it's not so easy currently still. So I think there's two phases. One phase to build up the animal facility and genotyping and starting to really maintain the colony with very precise system. And the second phase is to start setting up if you want to make your own models and what is that needs for. And for that, I think you should send somebody to our lab for 2 years to train. I think that's the only way. I mean, it takes at least 1 to 2 years to really be trained to understand. Of course, if you have somebody abroad, a PhD who could come to run it, so that would be the way, because it's so many things one has to learn. So it's a 2 years project, I would say.
Okay.
2 years.
That's what we are.
2 years.
It's a 2 years, yes.
Yeah.
Ari has been very,
Yeah,
I know, brave, I would say.
2 years. Yeah, Finland is Finland has been named as the happiest place in the world. So try it. Try.
Yeah, you are right. You have learned a lot. Yes.
Yeah. Okay.
But we are happy to support you. But I have been telling that to Ari that it's a lot of work, train, and it's handwork. It's not only brain power. It's a lot of, small things that somebody had to understand. I mean, have a touch in the fingers.
Oh, yeah.
I think it's different from pipetting in a lab, because if you have a good pipette, you always can take 5 microliters. Yeah. But making the mice, you have to feel the pipette.
Yes, okay. With the mood also, maybe, yeah. The good mood also.
I absolutely, yes. Okay. But I think when you get this colony in Indonesia, I think robust genotyping, and start to see that it's really in any day it works. So I think it's key.
Okay. Actually,
so again, I mean, if students sometimes think that PCR is boring, it's so easy, boring, but it's not. If you want to really know what's going on, so it's really challenging.
Okay. Yeah, Ari. Yeah. Actually, we would like to build this animal disease model, yeah, Ari. But the funding is not coming in the last years.
So we will buy first, Mati. We will buy first, just to initiate, the project. So that
I think that's a very good plan. That's what would be my suggestion.
Yeah.
Because
and then slowly build up it.
Yeah. Okay.
Okay, thank you for your suggestion, Prof. Mati Potanen. So another question.
Do you recognize this building behind me?
Yeah,
I mean, this painting. Yeah, it's my home office.
From the Bali, or I don't know.
I don't know. Ari would know.
Ari had gift.
Yeah, this is painting
present.
From yeah. Yeah. Okay. Bali. Yeah.
Bali, from Bali. Okay. So yeah.
Other question.
Other question for the student, from the student.
Yes, please.
Is there any questions from the students?
So here, there are also medical doctor here. Maybe it is interested also for the treatment. Yeah.
Especially working on endocrinology.
Yeah.
Prof. Mati, can I ask questions?
Yeah,
Yeah.
yeah.
This is actually very basic questions because there are many types of hydroxysorb dehydrogenase enzyme. What is your opinion at least in HSD17 beta? Is there any relations between one enzyme to another? I mean, is their work depend on another enzyme, or maybe they substitute another enzyme, or when one enzyme knock out, is there any relations with another enzyme, something like that? What is your opinion about their relations?
Yeah, I mean, I think there is, but we don't know what is the substituting enzymes. As I showed, we are now challenging the problem with the ovary, that even we are deleting the type 1 enzyme, we still have estradiol production. We expect that to be maybe type 7, maybe not. Same for the testis. In some cases, there is no compensatory system, so it seems to be that it depends. But this is actually what we are trying to now to understand and look which enzymes are substituting the other enzymes. So we are doing also some expression cloning now to try to understand, because this, as I said, the enzyme family, which I talked about, the whole nomenclature and definitions have came from in vitro studies. And I am always criticizing, it's my profession to criticize in vitro studies, but it's so easy to find, estradiol production from estrone, for example. If you incubate your cell culture for 3 days and let it be, of course something happens. But for the enzymology, the problem is that we don't have any more enzymologists around us. I remember when I started my studies at late 80s, there were some people who could discriminate in one cell 5 HSDs just because they were following the kinetics of the substrate. And nowadays, there's nobody who understands, in our lab at least, who understands enzyme kinetics. So everybody's doing just coding and expression, and I think we have lost a little bit, like related to your question, we have lost a little bit the expertise in basic biochemistry now on this new area of molecular biology. And we had to understand the enzymology and the kinetics. I mean, how the conversion rates will be, and that is there a possibility for this enzyme ever to see the substrate in vivo? Because in cell culture, you can just put a clean system and you put one compound and wait for 2 days, something happens. But what about in vivo when there are millions of compounds with different concentrations competing with the same system? So you have to have very, very high affinity for the substrate. And this is the challenge. And that's why we have been doing this in vivo models, even they're complicated, because many in vitro systems are too clean. But of course, I don't say that many in vitro models have been then led to very essential discoveries, of course. But we have to worry just and be aware about if your in vitro data doesn't correlate with in vivo. I'm just a little bit outside of your question, because years back there was a lot of people were doing reporter assays for gene activation. They put like reporters or promoter studies in vitro, and thousands of promoter studies were done in vitro, that you put different pieces of a promoter in front of the reporter gene and you translate them in cells and you see a lot of things. And we try to repeat these promoter studies in vivo by transgenic animals. We never could. We never could find, any of the in vitro promoter studies we couldn't replicate in vivo. Meaning that the whole regulatory regions in vivo is totally different from what people used at that time in their cell cultures. And that gave me a little bit like kind of biased for the in vitro studies. We tried so heavily to understand promoter regulation by using transgenic animals. And most of the case, nothing related to what we found in vitro. So this translation, I think, is very important. And that's why we still put a lot of focus in vivo studies. And then the next step, of course, is can we translate this to human biology and in clinics? So there's so many steps that it's a huge task, but it's a good question. What is compensating? And it's in most of the cases, it's still open in the cases that we have compensation. And that's what we try to do now and try to understand. Because of course we can make inhibitors, like we are now working with inhibitors that are hitting 2 of the enzymes at the same time. That if you have compensatory system, of course you can design an inhibitor that blocks both pathways. So in that sense, it's very important to understand the compensatory systems if you want to ever make a drug for if there's a compensation. This is true.
You showed a picture about abnormality in the mouse eye, indicating that this enzyme have a relation with this retinoid signaling pathway. So did you make a histological section? Is there any something abnormality in the histology?
No, we didn't. That's a good question. Again, we should have done, but again, it was going to the direction that we didn't want to go, so we never did. But yeah, there's a lot of interesting, preliminary data in our data files that we have never followed. The whole thing about even the placental dysfunction in that knockout. How is that related potential to retinoid signaling? Because retinoids are certainly involved in placental, regulating placental development. We haven't really had time to follow that, but we have the mice there, we have ideas, but even we have some money, we still have too little money to do all these things that would be really interesting. So we are suffering with the same problem currently in Finland, a little bit with the lack of academic funding on science, and we are not very happy with the current government that we have. But yeah, that's a good point that we had never followed that carefully.
Yeah. Thank you.
Thank you, Prof. From the students, is there any questions?
So here maybe I pointed for the question from the student. So siapa nih? Rosa Sylvia. Do you want to conduct the research with this female reproductive medicine? Ada nggak, Rosa Sylvia?
Ya, saya Prof. Mohon maaf. I'm sorry I can't show the video because the internet channel not so good.
Maybe you have a question for this nice lecture from Professor Mati. Yeah, because you are a medical doctor, and then we'll make a research in this female reproductive system. Yeah.
Yes, Prof.
Okay.
Thank you. Okay, actually, I am interested in the female reproductive endocrinology, and but since I work in the infertility field, and sometimes I found some patient with abnormal sperm quality, and they face infertility problem. Do you think is there any recommendation related to this basic knowledge to apply in the clinical field regarding the male infertility based on what you have done in your research? Any comment about this?
Not at the moment. This is a good question, and we are working constantly with our gynecology department with people who do IVFs, and I have a good friend and colleague who is leading one of the heads of the gyne department. and he is struggling with this problem all the time with the and actually he's doing ICSI, and if you want, I don't know if you do ICSI, but he's one of the persons who can go around the world and train how to pick a sperm from a testis. I think he has been somewhere in Asia sometimes, even train people. So he's one of the in Finland who actually are taking if they find some sperms in the testes, to do like testicular surgeries to find the sperms for ICSI. So he would be much better to answer your questions. Our basic results research now at this point really don't have any kind of solutions, even hypotheses for your problem. I think in our department, actually, there is a very strong group, not my group, working on spermatogenesis. And they are looking on different factors that are affecting spermatogenesis. But really currently not our field, even though when Ari was with us, we had a lot of work on epididymis and trying to look on post-testicular sperm maturation. But I have not really studied that for some years now. So unfortunately, I don't have any good suggestions to you. Only thing I can suggest you a good name that you can contact.
Okay, thank you.
So,
Thank you.
but one of the options, actually, what we HST is actually one of our ideas is. So we have been also working on the male hormonal contraception possibility, and there, of course, there could be a possibility for HSTs because this type 3 inhibition. Currently, we are thinking that could actually might lead, together with the current testosterone, progesterone implants, to further support the, drop down the spermatogenesis. So that's what we are a little bit following up. That could we use HST type 3 inhibitor as part of the concept for male hormonal contraception. Their academic, the hypothesis seems to be valid, and we are now testing that, actually trying to make a double knockout with the LS receptor and type 3 knockout. So, and that's a fascinating field. I don't know how it is in your country, but this is something we worked years back, trying to help men with hormonal contraceptives, because they are not, I think they are in Asia, of course, they are more effective, some Asian population. European population, it doesn't drop enough, the spermatogenesis, so it has not been really going through here. So,
yeah.
Okay, thank you very much, however.
Yeah. Okay.
Okay, is there still any questions? We still have 7 minutes.
Mm-hmm.
I have one more.
Ari.
Okay, so related to the previous question. So when you make a knockout for type 3 in male, did you observe the same phenotype with, for example, androgen receptor knockout mouse, for example?
It's not totally same, but it mimics, and there is now a paper going on that there is some androgen action left, and it's not so dramatic like a androgen receptor knockout. And of course, as I showed you, also there is androstenedione, there's some leakage of testosterone production, and that of course at the puberty it goes up. So if you have total lack of androgen receptor, so of course it's more severe phenotype for sure. So it's not as strong as it is with the AR knockout. But as I showed, it's very interesting that it very nicely mimics what we see in patients with type 3 mutations. So they are interesting patients actually, because they are really in Gaza region. They are, the incidence is not so low, and then they are really like boys who are raised up as girls often because they are not virilized normally, and then at the puberty they start to masculinize. And I think it's so now that in Gaza region they actually have they have a kind of they will test every baby to be sure that because it's good to be, treated early enough with surgery, whatever person they wants to, family wants to have, it depends, but yeah.
Okay. Thank you.
Okay. Is there still any question? Prof, I'm asking, but this is just filling my curiosity because I'm writing here what is the HE. In your experiments about HSD17 beta 7 knockout mice, you use 3 types of group, right? The knockout group, wild type group, and another one is HE. What is the HE, Prof?
What do we refer to?
The experiments about HSD17 beta 7 knockout mice. I'm sorry, but maybe I'm missing the informations there. You use 3 types of group, right? The knockout group.
Yeah, no, sorry, yeah, sure. I can even show if I can, but it's that, this is a good question for you also when you plan to use genetically modified mouse models. So we often use also heterozygotes, meaning that one allele is lost. In many cases, of course, it is so that one allele is enough to have normal physiology. But if in some genes you have a dose response actually, so that if you're losing one of the alleles, so you already have a small phenotype. Of course, you would expect that to be less than it is in the full knockout. But very often in the beginning, we use also heterozygous mice in phenotype to be see if there is a dose dependency on a phenotype. And if there is, so it's interesting. So as I showed you shortly, for the type 12, we have very strong phenotype already when we delete one of the alleles. So that would be maybe my suggestion, that also to in the beginning to look on also those potential changes that you find by, and especially that we have used that when we have a very strong phenotype that the animals are dying, let's say they don't grow at all, if you have both of the alleles missing. So then you can maybe look on is there a phenotype and you have dose, little bit dose, because you can also of course then, and what you should do is not just to look on the genotype but also look the messenger RNA levels. Because like in some cases, like I showed for the type 12, when you're losing one of the alleles, so the RNA goes to double. So basically there's no compensation for the other allele. Often it is so that even you have heterozygous mice, the messenger RNA is still normal level, so that the other allele is compensating it. But sometimes it doesn't. And if it doesn't, then you can show that, okay, messenger RNA is double, protein is double, and you have a phenotype, change in the physiology, then you can maybe make a conclusion that, okay, there is a dose response in this. And if there is, it's interesting. I think it's very good because I would say that gives also maybe, if you think about the clinical applications, if you have this kind of dose dependency in your knockout, so it might be indicate that pharmacologically it's interesting target, because with pharmacological inhibition you never get 100% inhibition. So likely, so it depends a little bit, but I think it's a good test often, and that's what it meant actually there. So that we have all the different combinations, 2 wild types, 2 knockouts, 1 wild type, 1 knockout. So yeah, good question, and good to take into account when you do phenotyping.
Okay, thank you, Prof. We already it's already 15:30. Thank you so much for your amazing lectures. Before we end this lecture, can we take a picture together?
Together, okay, yeah.
For the students, can you turn on your camera?
Please turn on your camera, please, student. So,
siapa yang lagi on ya, Mbak Nova? Bisa tolong difoto, Mbak?
Ya, boleh.
Nova?
Iya, Prof, boleh, Prof.
Buka ya. Pak Yulhasri, ini belum on. Yulhasri, sholat mungkin. So, another student. Pada kemana nih? Oke.
1, 2, 3.
So, okay, another. Three layers, ya, ada 3 ya.
Yeah. 1, 2, 3.
Okay.
Once again. One, 2, 3.
Okay.
Thank you.
Yeah, thank you for all, and special thanks for Professor Mati Potanen for the nice, amazing lecture today. And we meet next week, yeah, with the proposal examination from our student. Yeah, thank you.
Okay, thank you all.
Yeah, thank you, Mati. Thank you, Mati. Yeah, see you next week. See you next week.
Keep in touch.